for the last portion of this, we're going to do a fireside, and we've been talking a lot about hope, and the way in which we hear about progress in the field is often through the media. So as part of wanting to host a great event, we invited GEN team, who does super solid scientific coverage, as well as coverage of technology. Just a great group to help us cover this meeting. And Kevin Davies at GEN is also a long history of thinking about this space, including a recent work writing a book on gene editing. So excited to have Kevin moderate this fireside with George and I. And then from here, we'll break into the discussion, and then we'll bring it back at the end.
Fantastic. Hello, everybody. I'm Kevin. Great to be here. I'm the editorial director for GEN, Genetic Engineering and Biotechnology News. Bit of a mouthful. It's the venerable biotech publication that's been in existence for, I think, 45 years. I think more relevantly for today, I'm the author of a book that'll be coming out next year called, I won't reveal what it's called, but it's going to be a history and a biography of sickle cell disease. I think my one contribution was to introduce Eric to Victoria Gray, who until 10 minutes ago was sitting in the front row just soaking up all of this incredible information. Of course, she's become a very high-profile patient advocate for sickle cell disease, the first patient to receive Casgevy. She's the special guest of honor here at the GATC event, and you'll hear from her this afternoon. I think everyone here has heard snippets of her story, but you've not heard her story. You absolutely don't want to leave early. You'll want to stick around for that. But we have another special guest here, George Church. Of course, everybody knows George. I was laughing at in that introductory thing this morning. Can you find 3 people who don't know George Church? I highly doubt it. So he's graciously agreed to be here to take questions, and I've got a few lined up, but I'd love to keep at least 5 or 10 minutes for your questions. So please think of some interesting, probing, surprising questions. As Sonia said, she wants to be surprised. So, George, I don't know if you've been in counseling or talking to Eric about the inception of this GATC community. You've done these sorts of things before. You've had the Personal Genome Project and many others. I'm sure I've forgotten a whole bunch. What makes this one special? What gives you hope, and what should Eric and his colleagues be thinking of to really make this a sustainable and lasting and impactful community building initiative?
Yeah, I was really touched by one of the first comments from the audience earlier today, that this had the feeling of being more forward-looking, and we're all in it together to get to something that we all want, rather than we're just specialists in some field and we want to deliver what we did a few years ago. And that has a resonance with me with my other favorite meetings of all times, which is when we started the Genome Project in 1984 with Department of Energy, and then when we started the Brain Initiative in 2011-12. So, because they had that feeling that it seems almost impossible when you first talk about it, but the more you talk about it, the more reasonable and imperative it becomes. And I think genetic agency is one of those things. And so it's really great to be in this.
Yeah. There are 2 quotes on the website that I wanted to throw at you and get your response that really sort of encapsulate, I think, what the event is and this community is trying to do. The first is, we will need to overcome major challenges, technological and otherwise, for all of these treatments to become a reality. So in 2 minutes or less, what are the biggest challenges in your view?
Well, there are technical ones, which we've heard about a fair amount. But financial ones, in particular, when we're dealing with rare diseases, or diseases that involve very currently expensive reagents, and then social ones where people get educated as to what's available and what's possible. But I don't think that would normally be a problem, and that seems to have been a problem for getting genome sequencing going to the broad. But here, when you have a family that's in trouble, they in a surprising number of cases, they take the initiative. So, but I think the funding and the technology is still challenges.
An earlier speaker briefly mentioned the recent tragic patient death in the Intellia trial, and I think we're still trying to figure out exactly what was going on there. But it's a sobering reminder that, you know, for all of the progress we're making in gene therapy and gene editing, we still don't have all the answers. I wonder if you think we'll finally reach a point where we can run these trials without having to worry about immune responses or off targets or whatever is going on in this particular instance.
Well, I mean, I think it behooves us to bring down the dose as much as possible, and a lot of technology can be focused on that. We clearly have some successes. We have Victoria right here and baby KJ and so forth. And I think those involve lower doses. And so that's something we should definitely be aiming for. New vectors might be lower toxicity. And there's no shortage. I mean, there's no shortage of viral and non-viral vectors coming up through the pipelines. So, and some of them are, you know, due to AI and large libraries, and some of them are due to bioprospecting, and the list goes on. We're only limited by our ingenuity and hard work.
Eric, do you want to comment on the low dose?
It's a big motivation for our work, as I talked about earlier with Dyno. We're able to reach 80% of muscle fibers at 25 times lower dose than what's currently on the market for muscle gene therapies. So, like, there's a lot of new technologies that are in the works, and one of the reasons why I wanted to have this meeting is, despite all those challenges, you know, the things that are at that clinical stage right now, the underlying technologies that made up that drug, those were developed 5, maybe even 10 years ago. So even though we're seeing challenges today with what's in the clinic right now, there's still a great reason for optimism when we look ahead about, if this is working in this way, then what can we expect from the next generation? So to build some enthusiasm, some excitement, some hype, even from that next generation to drive that investment, I felt that was going to be, you know, one of the things that could come from this meeting.
George, the other comment in the run up to this meeting was the other quote that Eric put up on the website was, it requires a community working together at pace to turn powerful emerging technologies into patient impact. And you mentioned baby KJ, which of course was the dominant story at the beginning of this year, a fantastic example of public-private community coming together. Can that be a catalyst for other similar collaborations?
Well, absolutely. I mean, it's amazing how much faster a family will go than big pharma. I mean, we have a few examples at this meeting that I hope everybody hears. That's personal involvement. Sometimes they outsource scientists. Sometimes they become scientists in the process, like we heard today from Sonia. That's an incredibly powerful force.
Victoria's back with us. And of course, in 2013, George, you published one of the first examples of CRISPR gene editing in human cells. I wonder if you could just, I mean, we're so used to hearing you talk about technology, but I'd love to hear your thoughts about, you know, meeting Victoria, and not that you're trying to take credit for Casgevy, but you were there at the beginning, or pretty much the beginning, and I wonder what it's like to actually see the results of all of your work, and many people in this room, personified in the amazing clinical benefit that it's had in Victoria and dozens of other patients.
Well, it definitely is a team effort, and Victoria is part of the team, definitely. I think CRISPR was not our first shot on goal. It was our fifth, I think, technology, and then there's probably, I don't even know how to count the number of technologies since then. But it is very gratifying to see patients without any symptoms, or even with reduced symptoms. In this case, and also in the work that we've done with transplants that also use CRISPR to make 69 edits. Here, we're just trying to make one edit. Most of the things in this meeting are about making one edit, but there are other at CAR T, we make up to 6 edits. So yeah, it's just amazing to watch these people that inspire us.
Yeah, I'm just going to change tack slightly. You're sat next to Eric, of course, one of your many proteges. I'm just curious. You've launched dozens and dozens of companies. Where does Dyno sit amongst your the ranks of your favorites that have spun out of your lab?
Eric's giving me permission to say that he might be number 2.
You can tell him.
Tied for number 1. Let's put it that way.
Sorry?
I won't say how many he's tied with.
What makes a good founding CEO? We were talking quite recently about, you would never want to, and maybe were never offered the chance to sort of leave academia and go run one of these big ideas that spawned. But you've seen dozens of people, leave the lab, and some have flourished and some maybe not. What are maybe, and you may have some insightful advice for some of the people here, maybe we've heard this morning, who are following in those sorts of footsteps.
I'm not sure that kind of insightful or advice is my forte, but initially I felt that not only did my postdocs not qualify my mind to be CEOs, then I even start a company, but then Francois Vignon starting Advitro and Luhan Yang starting eGenesis completely changed my mind on that. And they not only were capable of co-founding, but of actually being CEOs. And Eric is another perfect example of someone who, none of these 3 had training in business school as far as I knew, or any of the management or anything, but they all rose to the occasion in a big way. So I don't think it's obvious that everybody can do that, but I think you know it, and if you don't like it, then you can always step aside and find a better CEO.
Yeah.
But that hasn't been the case for most of my startups. They really are postdocs that just move right in.
Yeah.
I could say one thing
on
on how George helped me. I guess 2 things. One is George is like a magnet, right? People hear about the success of the lab, they want to come. They also know that given everything George has worked on, George has got a pretty open mind. So if they've got some far out idea, that's going to be a good place to go to make it real. So I did that with George and joined the lab. I was actually surprised to see so many other entrepreneurially minded folks, like Francois, Luhan that he mentioned, but even when I was in the lab, overlapping with 5-10 other folks thinking about startups. So like you kind of catch the enthusiasm from them, learn from others who are a little bit ahead. And that, I think, trend has continued. So that was definitely one thing that really helped, was just seeing the examples of others who've come just before and learning from that. And the second thing about George is he's so open-minded. I don't think he's ever said no. So you can tell him a crazy idea. Even I think I want to be the CEO. I think I'm the right person for the job.
Yep.
And
yes,
and George will find a way to kind of push you in the right direction, or even maybe help you to think bigger. And so like that initial support is a big step towards and finally finding a way to make it happen.
Great.
Thanks, George.
thank you.
Any questions from the audience for George or Eric? George, go ahead. Yes, please.
So, my name is Daniel Lazette. I'm the Justice Officer at Theater. There's been a lot of discussion today about therapies faster and cheaper, not just as like an incremental scale, but it's like fundamental level to that there. And I can't help but thinking 2 weeks ago when I went to China, getting sense of that physics, and the Chinese biotech ecosystem moves at, on average, probably 3x faster than the US ecosystem, probably for 90% less amount. Like meat, web, cost, all modalities, all. But we haven't talked about that ecosystem yet today in the session side. So I'd love for you guys to think a little bit about whether that sort of changes your view on what's possible, and how that sort of shifts your thinking about where the policy community and the share really kind of work together.
Yeah, so I've co-founded some companies in China. For example, Kihan, which is in Hangzhou, Luhan, who helped us start CRISPR and helped us start eGenesis. And it is impressive what they can do, especially the IIT system, which is something I think we could learn a little bit from. We've already moved one drug for CAR T that has 6 edits in it through the IIT system, 20 patients tested, and now we've got it approved in the United States for another 20 patients. So they actually can work together. That's a nice example where you can take advantage of the strength of the IIT system and accelerate the process of getting FDA approval. I think there are many others, and hopefully since this is a community building exercise, hopefully that community is more unstoppable than any government that would want to stop us.
Maybe I can just say one thing about that. Well, first off, to point out, there's going to be a roundtable on this. I think I saw a topic about this, so go join that if you're interested. I also agree with George. I think this is going to be a combination of both cooperation, partnership, and competition. So obviously, lots of opportunities for partnership as we're talking about here. On the competition side, we're talking about all this technology, much of which is being innovated, for example, here in the US. And so being able to find the opportunity to translate that quickly, to be an early adopter and then to turn that into real results, I think that's our game to lose, but it's going to take a lot in order for us to get there. There's the regulation side. Are there things we could simplify, streamline in order to similarly have a fast path to the clinic? And it seems like there's encouraging steps there. The plausible mechanism pathway and other things that are underway, but we need to kind of realize the benefits that have been imagined through that by bringing that into real action. And then the other side of it with China, I've spent a lot of time there. It's also people that work really hard. And so it's going to take a lot of hard work for us to compete, but I think there's a really good reason to go after that. There's a great future we want to build. There's many patients who want to be part of that, who need those therapies. And so being willing to make that commitment and find a way to put the time in to solve these problems together, I think that's also going to be really key.
I'm going to just pivot off of China since you brought it up. It just came, 7 years ago, George, we had the whole CRISPR baby saga, and you were one of very few scientists who were willing to give He Jiankui a little bit of the benefit of the doubt. There was a great quote in Science where I think you said something, well, was he guilty of some sloppy paperwork or something like that? That probably wasn't taken in the full context, but now it seems we're talking about genetic intervention here, and obviously we're focusing on somatic intervention, but it seems like germline is starting to, there are some companies now, one in New York, Manhattan Genomics, with one of your former colleagues, Ariona, is a co-founder of that company, I believe. What are your thoughts about seeing this coming back into the marketplace?
Yeah. Well, so there are a few things that are still missing from it. It is that there's not a really clear case where it works and nothing else does, or even on paper should work when nothing else does. If it's a late onset disease or even medium onset, the clinical trials are going to be very long. And then the third thing is that 8.1 billion of us have missed the boat on that one, and so we have no genetic agency for ourselves. We can act on behalf of the next generation as proxy.
Yeah.
But it's a tiny fraction of the population. So I think those are three kind of showstoppers for now.
Yeah.
But I banned the word impossible from my lab, so
yes,
wait and see.
You want to be supportive. Yeah. Well, that's great for lunch. But Eric, I know you've got you're speaking to George right after lunch, correct? On AI.
George will give the talk, and then we want to have more Q&A. So let's break now. Conversation. Let's just bring that around again. Speakers are going to facilitate that. I'll call on folks just at the end to say a few words about what you thought was interesting, and then we'll again head out for lunch.
Thank you very much. Thank you, everyone.